Welcome to this overview of Roche 454 sequencing, where we unpack the pyrosequencing platform and its technical foundations. In the next few minutes, I'll guide you through the core concepts, workflows, and the practical implications of long-read pyrosequencing, using the diagrams and illustrations on each slide to make the process easy to follow and intuitive. This slide highlights early milestones achieved with the 454 platform, from the first individual human genome sequencing to reconstructing ancient DNA and enabling rapid microbial outbreak tracking. The icons and timeline emphasize how long-read pyrosequencing proved essential for assembling complex genomes and recovering degraded historical samples, establishing its unique value in de novo assembly and paleogenomics. Here we walk through the four-stage 454 workflow at a high level. Universal library preparation, emulsion PCR amplification, pico-titer plate loading, and sequencing by synthesis. The sequence of panels shows how fragmented DNA is converted into millions of individually amplified beads, how those beads are loaded into tiny wells, and finally, how sequential nucleotide flows generate light signals for readout. This page breaks down library preparation and bead capture. Genomic DNA is sheared, adapters are ligated, single strands are generated, and each fragment is captured onto a primer-coated bead as a clonal seed. The illustrations of fragments and beads show how, ideally, one template per bead initiates clonal amplification, setting the stage for accurate downstream sequencing. The emulsion PCR slide explains how single DNA-bound beads are incorporated into water in oil droplets to create isolated microreactors. Thermal cycling then amplifies each template clonally, producing roughly 1 million copies per bead, while isolation prevents cross-contamination between beads and ensures each well will represent one distinct clonal population for sequencing. This process flow diagram follows what happens after amplification. The emulsion is broken, and the beads are recovered. Magnetic enrichment concentrates DNA-bearing beads. They are deposited into pico-titer plate wells, and enzyme beads are paired with each DNA bead to position the clonal populations for pyrosequencing. The visuals convey the physical steps that link amplification to signal generation. The pyrosequencing reaction cycle is shown step by step. Individual nucleotides are flowed cyclically. Incorporation releases inorganic pyrophosphate, and an enzymatic cascade converts that PPI into ATP and then light via luciferase. CCD detectors quantify the light, and signal intensity correlates with the number of identical bases incorporated, producing the raw data used to infer sequence. Signal processing converts raw luminescence into base calls using real-time FPGA processing and flowgram generation. The processor translates peak heights to homopolymer lengths, and calibration with a TSIAG key corrects phasing across millions of wells, allowing accurate base assignment from noisy light signals through computational normalization and pattern recognition. The titanium chemistry slide summarizes performance metrics and the contiguity advantage. Modal read lengths around 500 bases. Q20 reads near 400 bases. Roughly 1 million reads per run, and 350,500 megabases of data output in a 9-hour run. The longer reads improve contiguity in assembly diagrams, showing clearer overlaps and fewer gaps compared with short-read platforms. This slide balances advantages and limitations. 454 offered long reads, useful for de novo assembly, spanning repeats, and detecting structural variants. But it also had higher reagent costs, lower throughput compared with short-read competitors, and challenges calling long homopolymers, which could lead to insertion-deletion errors for runs longer than about 8 bases. The application slide emphasizes impact across paleogenomics and metagenomics, the establishment of NGS experimental and bioinformatics standards, and the critical role of long-read information for resolving repetitive regions and improving assembly quality. The three panels show how 454 data-informed experimental design, analysis pipelines, and benchmarks that influence the broader next-generation sequencing field. The final slide covers cessation and legacy. Production wound down in the mid-2010s as cheaper short-read and emerging single-molecule platforms gained dominance. Yet 454's proof of concept for massively parallel pyrosequencing informed successor technologies. Its foundational innovations and lessons shaped later high-throughput sequencing platforms and left a lasting impact on genomics methodology.